Okay, so now first of all we'll be talking about the plasma membrane. What we'll be talking about the plasma membrane. So we know that first of all, if you draw a cell like small or just small schematic like this, suppose this is a cell, and suppose we are actually drawing, this is suppose the plasma membrane. This is actually the, suppose this is the plasma membrane. So you can see that this plasma membrane, what is the important role? If we say what is the important role of a plasma membrane, point number one will be, it will create, this plasma membrane will create a permeability barrier. It will create what? A permeability barrier. How? Suppose inside the cell, in the cytosol, suppose if there is a pH is, suppose it is 7.4 and outside it is 7.2. So there is a difference in pH outside the cell and inside the cell, and this barrier, actually this membrane is actually creating a permeability barrier, so there is no mix and match between the fluid having different pHs. So point number one will be plasma membrane will act as a permeability barrier. Second, now you can also see that plasma membrane, they are also responsible for different kinds of regulated transport. They are also responsible for different kinds of regulated transport, right? If you want to transport from one part to another part, so the plasma membrane is very much essential. So there are different transporters present over the plasma membrane which actually transport many important things. Suppose there are different transporters like this, there are several transporters like different, like Na plus, K plus pump, all these kind of transporters are present. So suppose if you want to regulate a particular transporter, so you actually have different gradients. We will be discussing with the transport in the later classes. So yeah, we are having different transporters which are actually helping in the regulated transport, and for that regulated transport, plasma membrane is very much essential, that is also important. Now another important thing is that, yes, suppose this is a cell, okay. Now you can also see that in the membrane we are also having different kinds of receptors. And different ligands are binding over here, and for that different signal transductions are actually taking place within the cell. So in the plasma membrane several receptors are also present, and when they are binding with the ligands, they are actually introducing several amounts of signal transduction. So the third important function will be also the signal transduction. And I'll suggest you everybody to write what I'm writing. That is very important. You have to make your notes like this. Okay. So signal transduction is another function. Okay. Now suppose we are having a cell. This is one cell, this is another cell, and this, suppose this is the plasma membrane, and there are certain different kinds of cell adhesion molecules are present. These cell adhesion molecules actually interact with the other cell adhesion molecules of the other cell like this. They will interact with this like this, and by the help of these cell adhesion molecules are actually linked with the plasma membrane, and these two cell adhesion molecules interact with each other. What are they? These are nothing but these are the CAM. These are the cell adhesion molecules. What? They are the cell adhesion molecules. So you can see that in the plasma membrane there are several cell adhesion molecules present which actually interact with the other cell adhesion molecules linked with the plasma membrane of the another cell, and as a result of that you can have cell interactions. So another function of the plasma membrane will be what? It will be cell interactions. There will be cell adhesions, all these kind of things. Now, another important thing, fifth. Okay, there are certain molecules which are also, this plasma membrane is also very much important to maintain. They are also very much important to maintain the cell shape. Okay, we will study that basically there are different cytoskeletons present in the cell, like there are actin filaments, okay, there are intermediate filaments, all these presents. These kind of cytoskeleton, actually they are linked with the membrane by help of certain proteins, and they are actually maintain the overall shape of the cells. So actually different proteins are linked with the plasma membrane, and these different proteins interact with those cytoskeleton membrane, and they basically help to maintain the shape of the cell. Cell interaction, yes, cell interactions. Now, sixth point, what are the other functions? Can you tell me another function? Can you tell me another function? Just write it in the comment box. I have given you 5. Just tell me one more. Use the chat box to comment your answer. You can use the chat box. So it acts as a permeability barrier, it acts as a regulated transport, there is a signal transduction, there is cell interactions, there is transport of molecules inside and outside the cell. So yeah, we have talked about this, right? Regulated transport, inside or outside. So this has been talked. Signal transduction, cell interactions, maintaining the cell shapes. We can also write one important thing, that there are several kinds of enzymes present in the plasma membrane. There are several enzymes are present in the plasma membrane. Suppose in case of the bacterial plasma membrane, there are huge amount of enzymes which are important for your respiration, bacterial respirations. And we actually know that from the endosymbiotic theory, we know that the mitochondria is actually a symbiont of the protobacteria, and what enzyme present in the plasma membrane of the bacteria is also present in the inner membrane of the mitochondria. So we will be dealing with all those concepts, so you don't have to worry about it. But as a result of this, membrane bound enzyme, very good Vibhash. Hold the contents like cell organelles inside, very good Priti. Blood groups, okay, fine, this is only specific of the RBC membrane, fine, it's also correct. There are different proteins which are different glycosylated, and different blood groups are okay identified. Now, so from these 6 functions, we can tell one thing, is that this plasma membranes have one thing. They have both structural and functional role. They have both structural as well as functional role. Contain cell surface receptors, yes Priya, because we have also mentioned in the signal transduction cell surface receptors. Shumanapal is telling regulates the molecular traffics across the boundary. Yes, traffic transport, that is the regulated transport, right? Very good. Now everybody going good. So now I am just going to the next portion. Now see this very carefully. We actually study about the plasma membrane, okay, but for that we take the model system as the mammalian mature RBC. We take the model system as the mammalian mature RBC. Can anybody tell me why? In order to study the plasma membrane, we actually take the model system as the mammalian mature RBC. But why we actually take this mammalian mature RBC as a model system? In order to study the plasma membrane. Any. It's an open-ended class. Any point of view is welcomed. Anybody? It does not have nucleus, mitochondria and Golgi-like organelles. Absence of nucleus. Okay, fine. You're very good. But now relate it. Why it is actually used as to study the plasma membrane? You are going good. Easy to isolate. No, there are other cells. We can actually isolate many things. Okay. So easy to isolate will be a vague answer. Okay, Vibhas. Preeti is saying that presence or absence of antigens. Okay. So it's fine now. If we actually find some antigens, it will help us to actually study the plasma membrane in a better way. Flexibility. So how flexibility plays an important role to study the plasma membrane? So she is telling that all the functions are done by the plasma membrane. As there are no nucleus and the internal membranes, it will be easy to isolate. Yes, very good. That will be the reason. So suppose if we want to, if we take a particular, suppose hepatic cell, okay. So for that, suppose in other kinds of cell, you are having plasma membrane, you are having mitochondrial membrane, you are having chloroplast membrane, okay. You are having lysosomal membrane. So these are all unique membranes, and this is your plasma membrane. So now, in order to have only the plasma membrane, if you take mammalian mature RBC, the answer will be, as the RBC cell is not contaminated with other organelle membranes. Why? Because, write the answer. Because as, sorry, one second. It will be, as the RBC cell is not contaminated with other organelle membranes. It has only and only the plasma membrane with other organelle membrane, because it doesn't have any other organelles as well as the nucleus. Other organelle membranes. So the only membrane we will be getting is your plasma membrane. Okay, so all the membranes you will be derived from the RBC. All the membranes which you will be deriving from the RBC, all the membranes are derived from in RBC, which we will be getting are derived from plasma membrane only. From plasma membrane only. Is it clear? Is it clear? I think this section is clear, everybody? Very good. So everybody try to answer. Okay, everybody try to interact with me. That will be helpful for all of us. Now, let's go to another section. Given heading, chemical composition of membranes. Chemical composition of membranes. Chemical composition of membranes. Please explain once more. Yes, sure. Say this very carefully. Why we actually use the mammalian mature RBC as the model system in order to study the plasma membrane? Suppose this is an RBC, okay, and this is a normal cell. So in the normal cell, you are having plasma membrane, you are having mitochondria and mitochondrial membrane, you are having lysosome and lysosomal membrane, you are having peroxisomal membrane. So all the membranes of the organelles are also present. Now, if you want to isolate only the plasma membrane, it will be very much tough for you, because you will be having a mixture of all membranes, like plasma membrane, along with that you are having the different membranes of the organelles. But you just only want to study the plasma membrane. In RBC, you are having only the plasma membrane and there is no nucleus, there is only hemoglobin present. So actually the membrane which we are only getting that is nothing but your plasma membrane. So that's why mammalian mature RBC, not immature RBC, because in immature RBC there is nucleus, but in mature RBC there is no nucleus and other organelles. If you are having nucleus, then you are having nuclear membrane, so there will be again a contamination of the other membranes. But you want only and only plasma membrane. Is it clear, Priya? Very good. Now, chemical composition of the membranes. Chemical composition of the membranes. Suppose we are talking in case of RBCs. Okay, write in this manner: lipid, proteins, and carbohydrates. Lipid, proteins, and carbohydrates. In case of RBC, 45%. In case of RBC, 45% is lipids, about 40 to 47% is your proteins, and less than 8% will be your carbohydrates. So actually in several interviews they ask question like that: which kind of cells have the similar amount of lipid protein ratio, 1:1 ratio. So most of the cells, you can say that most of the cells, they actually have 1:1 lipid to protein ratio. So now there are several interviews, they will be asking you another kind of question, that so now tell me which kind of cell have lesser amount of lipids and higher amount of proteins. Why is there difference in the ratio? Yes, I'll be telling you, don't worry. Yes, most of the cells having this 1:1 ratio. Now why? Now we are talking about bacterial cell. We are talking about bacterial cell or bacterial plasma membrane. So it is bacterial plasma membrane, support, like this. And this is RBC plasma membrane. See this very carefully. In bacterial plasma membrane, lipid amount is about 30%, protein amount is 70%, and very less than 1%, if it is present, will be carbohydrates. Now why? To see this, why the amount of protein is so high in case of the bacterial plasma membrane, but is much less in other cells or the other eukaryotic cells. Why it is happening so? Now see this very carefully. What is happening? The thing is bacteria, peptidoglycan, okay, fine, but why? Now the thing is, listen very carefully. Okay, and peptidoglycan is the cell wall, okay, it's not the membrane. Bacteria does not have any mitochondria, okay, so it has to perform the function of the mitochondria by the help of their plasma membrane. So you have studied in your +2 levels that in case of the bacteria, they are having several kind of invaginations which are known as the mesosomes, and these are the invaginations of the plasma membrane. These are called what? These are known as the mesosomes. These are the invaginations of the plasma membrane, and as we know that these mesosomes actually help in the respiration of the bacteria. They actually help in the respiration of the bacteria. Now what is happening? The thing is, what function the inner membrane of the mitochondria is actually performing for the respiration, that function is being performed by the plasma membrane. For that you need bloody a lot of amount of proteins in the plasma membrane. Yes. So for your respiration you need huge amount of enzymes, and enzymes, maximum enzymes are proteins, and these are all present in the plasma membrane. So as bacteria, please write it down, as bacteria does not have the mitochondria, it has to perform the function of the mitochondria. Okay, it has to perform the function of mitochondria. Please write it down. Okay, now we know that the mitochondrial inner membrane has a lots of proteins, like different enzymes. All the membrane bound enzymes are the proteins. So for that purpose, bacterial membrane also consists of higher amount of proteins. Bacterial membrane consists of higher amount of proteins and transporters. Yes, I'll be repeating. See this very carefully. Bacteria does not have any mitochondria. So it has to perform the function of the mitochondria by the help of their membrane. This invagination structure of the membrane is called the mesosomes. Now we know that the mitochondrial inner membrane has a lots of protein which actually mainly perform the electron transport system. Okay. So for that purpose, bacterial membrane actually, as they are performing these kind of functions which are important for the respiration, they are having huge amount or higher amount of proteins than that of the lipids. So that's why in the bacterial membrane you can see 70% protein and 30% lipids. So most of the enzymes are actually present in the plasma membrane. So the proteins ratio rises up. It's also very important concept. Okay. Another important thing. That was about bacterial plasma membrane. Now in case of myelin sheath, lipid amount is 70%, 30% is protein and very less than 1% is carbohydrates. Why? Because they need a lot amount of insulation. Myelin sheath is a part of the neuron and they should be protected by any kind of shocks. So they need a lot more amount of insulation, and that's why the lipid profile or the lipid content in the myelin sheath is much higher. Very good, Vivash. Yeah, you're right. Is it clear? So basically, different cells, like in neuron cells, the lipid is to protein ratio is different than that of the RBC. The bacterial cell lipid is to protein ratio is different than that of the other cells, eukaryotic cells. So you can't tell that all cells have the same lipid is to protein ratio. No, it actually depends upon different functions. Right? So up to this, is it clear to everybody? Then we can move on to the next section, that is the membrane lipids. Please tell me, everybody. I want everybody's answer. Okay. Very good. So now let's move on to the next section. And I think you are writing all which I am writing in the board, right? So please make your notes. That's very important, because those who are going to join in this sales program, so the class, my class again from the next week will be after today's class. So it will be a continuation course, right? So what I am finishing today, after that in the first week of August, the first class of mine will be continued from today's class only. Okay, where I will be ending the class, from that I will be starting the next class. Now give heading membrane lipids. It's a very important concept. Huge questions are going to come from this section. Huge questions. And there is a very much relation with the biochemistry, but we will not go to the structure. We will be dealing the structure in the biochemistry, but we will go all the functional analysis in this cell biology portion. So now see this very carefully. This membrane lipids has a major role creating a permeability barrier. Suppose this is the membrane, these are the lipids, these are all the lipids. Okay, and we know that lipids are actually non-polar, and all the interactions which are taking place inside the lipids, this is the hydrophobic interactions. So these molecules actually can't go there, these molecules cannot come here. So they are actually creating a permeability barrier. Anything you have to go through a particular transporter by using energy or without energy, that is a different thing. But this lipid layer is actually creating the permeability barrier. So these lipids are mainly actually responsible for creating the permeability barrier of the membrane. Okay. Now, membrane lipids, second point, membrane lipids are also responsible for, they are also having a structural role. We will be dealing with this, what is the structural role. So they are also having a structural role. Cool. Very good. Now, we will be actually classifying these membrane lipids into two sections. What are they? One is glycerol based membrane lipids. Okay. Glycerol based membrane lipids, and one is sphingosine based. Yeah, glycolipids will be coming, but one is glycerol based. This is the broader actually classification. Sphingosine based membrane lipids. Now in glycerol based membrane lipids, there are glycerol based phospholipids. They are most common. And there are also some glycerol based glycolipids. Very less in amount, but there are. I'll be telling you. There are sphingosine based phospholipids, and there are sphingosine based glycolipids, and this is also most common. Okay, so everybody noted down this chart. Note down this chart. Yeah, please respond that you have noted down. Noted. Please respond noted. Good. Very good. Now, let's go to the first section. We'll go with this. Okay. Now, very good. चलो. So देखते हैं. Let's go to the first part. Glycerol based phospholipids. This section which I am actually going to teach you, these sections are very important for your exam point of view. First phospholipid which we will be dealing is first phosphatidyl ethanolamine. What? Phosphatidyl ethanolamine, which is written in what? PE. Okay. Point number 1, which is very important, is that one, this is the most abundant lipid. Most abundant lipid in bacteria. This is the most abundant lipid in bacteria. बहुत सारे questions इस part से आते हैं, बहुत. So many questions actually come from this section. So just make a highlight. Most abundant lipid in bacteria. Okay, next point. Second, Here, this lipid is mainly present. This lipid is mainly present on the cytosolic phase of the membrane. Suppose this is the membrane. This is your membrane. This is your exoplasm or outside the cell, and this is the cytosol. Now this phosphatidyl ethanolamine will be present like this. Sorry, one second. It will be present like this. One sec. On the cytosolic phase, majorly in the cytosolic phase. So you can see that the main chunk is present on the cytosolic phase like this. So we will write PE over here. Next, at pH 7, okay, net charge will be 0. Why? Because it is phosphatidyl ethanolamine, meaning there is a phosphate, so we will have a O minus, and ethanolamine will go to the structure in biochemistry. We will see that it will having this. So in at pH 7, so there will be equal minus and equal positive. So the net charge will be 0 at pH 7. And this phosphatidyl ethanolamine is actually referred to as, its common name is, there are certain exams like JNU actually ask questions in the name of common name. Common name is cephalin. What? Why is it so? Any particular reason? Yeah, I will be telling you all these things. Why it is present in the I will be telling you. Don't worry about it. Common name is cephalin. Okay. I'll be telling you everything. Just wait for a few minutes. Next type of phospholipid is phosphatidyl choline. Phosphatidyl choline. In short, we can write it PC. So they are having a group like this, CH3, CH3, CH3. So we will go to the structure in biochemistry, but this group is present. This group is present. And also there is a phosphate group like this. So we will be having all these kind of groups are present. So actually tell me, so what will be the charge at pH equal to 7? What will be the charge? What will be its charge at pH 7? What will be the net charge? Sorry. 0. Very good. So at pH 7 the net charge will be 0. Simple, like that of the ethanolamine, there was 3H, there are 3 CH3. Now phosphatidyl choline is abundant in RBC along with PE. PE is also abundant in RBC. So यहाँ पर भी लिख लो, it is most abundant lipid in bacteria and also in RBC. Okay, also in RBC, and same, just write over here the phosphatidyl choline. It is also abundant in RBC along with phosphatidyl ethanolamine. Now this PC, this is seen in the exoplasmic phase. Like, or phase or site. So suppose this is the membrane, so they will be present like this. This is the membrane, suppose this is the membrane, and this will be PC. Is very important. This PC, phosphatidylcholine, is absent in bacteria. It's absent in bacteria. Okay. So have you written this? Have you written this? Very good. So one question in BHU came, what is DPPC? Full form of DPPC is, we will be actually talking about all these things in biochemistry, like this is dipalmityl phosphatidylcholine, phosphatidylcholine. So what is DPPC? It is nothing but a surfactant present in, this is actually acts as a, they act as a surfactant, in the lungs. I think you have learned about this in your class 2 level also. DPPC is the dipalmityl phosphatidylcholine. So here is also, so you can see that this phospholipid is also present along with this along with 2 palmityl fatty acids. So they actually form this, yes, and that is also, that's why phosphatidylcholine, yes, you are right, that's why phosphatidylcholine is referred to as lecithin. PE was, phosphatidylethanolamine was called as the cephalin. PC is called as, or is known as the lecithin, right? Next. Now write phosphatidylserine. Phosphatidylserine, PS. Now as there is serine, there will be, there is phosphate, and there will be a O minus, like OH and O minus. So at pH7, its net charge will be negative. Net charge is negative due to this O minus, Okay. Yes, Shumanapal, you are right. This phosphatidylserine is absent in bacteria. This phosphatidylserine is absent in bacteria, and this phosphatidylserine is present in the cytosolic phase. It is present in the cytosolic phase of the membrane. So we can write in this manner, like if this is the membrane, so they are also present in, suppose this is the membrane, so they are present like same as that of the phosphatidylethanolamine. Okay, so PC was only at the exoplasmic end or exoplasmic phase. So this is PS. Next phospholipid is phosphatidyl inositol, PI. Now this is very important. Why it is very important? I am telling you. Okay, PI. Its net charge at pH 7 is also negative. At pH 7, it is also negative. Point number 1. This is also present at the cytosolic phase. Like this. This is PI. Now, what is another most important function of phosphatidyl inositol? What is that? They are involved in cell signaling. What? They are involved in cell signaling. They are involved in what? They are involved in cell signaling. How? Tell me. From this phosphatidyl inositol, if we add 2-phosphate, we are getting PIP2. We are getting what? PIP2. And if you add another phosphate, if you add 2-phosphate, you will get PIP2. If you add another phosphate, you will get PIP3. Okay, now from PIP2 it can be degraded, it can be actually differentiated into two forms like DAG, common name of phosphatidylserine lipoamino acid. Yeah, yes, you are going good. Yes, calcium release pathway. Yes, very good. But what I'm going to tell you that DAG IP3. So PIP3, okay, then DAG IP3, they actually act as the secondary messenger in your cell signaling. They act as the secondary messenger. What are their functions? Yeah, obviously they are actually involved in the calcium A pathway. DAG is a different pathway. PIP has a different pathway. All these things are, that is fine. But PIP3, DAG, IP3, these are all the secondary messengers. So we can actually tell this phosphatidyl inositol. Phosphatidyl inositol is a precursor. Is what? Is a precursor for secondary messenger. It's a question of JNU CWB 2013. So precursor for secondary messenger is PI, phosphatidyl inositol, which is actually present in the cytosolic domain. Why? Because actually you need to do the cell signaling within the cell. That's why you need this kind of phospholipids in the cytosolic domain. You need those kind of phospholipids on the exoplasmic domain when you are actually acting as a receptors. When you are actually doing particular cell signaling work, you need your phospholipids to face the cytosolic domains. So that is the main idea regarding that different phospholipids present at different domains. So they are having different functions. Now, another important thing, which is that many books actually don't write this, but actually questions do come from this portion. Fifth, that is phosphatidyl glycerol. Phosphatidyl glycerol and diphosphatidyl glycerol. Phosphatidyl glycerol and diphosphatidyl glycerol. This phosphatidyl glycerol is called the cardiolipin. It's very important. Now, what I'm going to write is also very important, is that these are actually phosphatidyl glycerol and cardiolipin or the diphosphatidyl glycerol. These are actually the bacterial membrane lipids along with phosphatidyl ethylenamine, which we have discussed in the first part. So these are actually the bacterial membrane lipids. This is also very important, very important. So which kind of membrane lipids are found in bacteria? So you will tell PE, PG and DPG, and this is called cardiolipin. This question has been asked in HU. Okay, these things are also very important. Now, if the same question asked in a different manner, so which membrane consists higher amount of cardiolipin in eukaryotes? What will you tell? Question: which membrane consists of higher amount of cardiolipin in eukaryotes? What will be your answer? Which membrane? Which mitochondrial membrane? Yes, because I have explained you that what is the membrane of the bacteria, that is exactly same composition of the membrane is present in the inner membrane of your mitochondria. So in the inner membrane of your mitochondria, yes, everybody correct, yes, I am going to repeat it. See this very carefully. I told you one thing is that phosphatidyl glycerol and diphosphatidyl glycerol, these two actually membrane lipids are present in huge amount in the bacterial membranes, right? So if a question asks in this manner, We have learned what? We have learned that PE, phosphatidylethylamine, phosphatidylglycerol, and diphosphatidyl which is called the cardiolipin, they are present in huge amount in the bacterial membrane lipids. So if a question asks in such a manner that which membrane lipids or which membrane consists of higher amount of cardiolipin in eukaryotes, so which is actually the similar kind of membrane than like that of the bacteria, that is the mitochondrial inner membrane, because we have discussed that the mitochondrial inner membrane and the bacterial membrane are the same thing. They are having same lipid to profile ratio and they are having same kinds of membrane lipids. So what is present in your bacterial membrane, that same composition will be present in your mitochondrial inner membrane. Yeah, now that is also a very important concept. Yeah, actually mitochondria has come from a different kind of protobacteria. Chloroplast has also come from a different kind of protobacteria. So yeah, actually if you're seeing those kind of membranes in such kind of protobacteria from where the endosymbiosis has been formed to form the chloroplast, you are actually going to see same composition in the chloroplast membrane too. Yes, that is also correct. So you can majorly, we are talking about the animal cells and we will be mainly discussing about the mitochondria, okay, and all the cells which I am talking about mainly they are about the animal cells. We are not going into plant cells. That's why mitochondria, not otherwise it can be of chloroplast. Well, very good. Let's go to another deeper level. Let's go to a deeper level. See this very carefully. So we can say that from bacteria or the bacteria to eukaryotes, okay, many phospholipids are present, like phosphatidyllethylamine. These are conserved to eukaryotes as well. Now see this very carefully. In all of them, the phospholipids which are present, phospholipids, you know that what are phospholipids? These are glycerol plus fatty acids. These are what? Glycerol plus fatty acid plus phosphate group, and along with that ethanolamine group can be present, choline group can be present, all these kind of things can be present. Now the thing which are very important is that the glycerol in the phospholipids present in from eubacteria to eukaryote, all the glycerol are present in D form, as it has been, because glycerol comes from glyceraldehyde. So it is derived from D-glyceraldehyde. Okay. So from eubacteria to eukaryotes is very important question. All the glycerol in the fatty acids are in the D form. All the glycerol in the fatty acids are in what? D form. Very, very important. And this glycerol actually bind with the fatty acids by the help of ester bonds. This kind of bond is called ester bond. This is called the COOR is bond called, this bond is known as the ester bonds. Okay. Now exceptions are there. Exceptions are also there. In eukaryotes, there are certain kind of membrane lipids or the membrane phospholipids called the plasmalogens. Certain membrane lipids, these are also membrane lipids, and these plasmalogens, these are also the membrane phospholipids, but they are having ether bonds at C1 carbon. That is an exception you have to know and remember. Many entrance examination actually ask this type of questions. So they are having what bond? They are having ether bonds at C1. So which exception is very important? Can you repeat the D form? D form is nothing. D form actually we have, you know that there are D form and L form in the sugars. Glyceraldehyde is a sugar, 3 carbon sugar, right? It is a triose. So from that triose D sugar, your D glycerol has been derived, D form. Okay, in actually is a part of the stereochemistry. D and L form, and optical there is small d and small l form, right? So D and L form, so it has been derived from the D sugar, D glyceraldehyde. So those glycerol which are actually derived from the D glyceraldehyde, they are called D glycerol. So D form of glycerol actually present in the eubacteria to eukaryotes. No L form is present. Any particular reason? First of all, that will be capital D. Okay, small d and small l is different. That is dextro and levorotatory. And capital D and capital L are stereochemical figures. Now there are, first of all, it has been observed. There is no particular reason found why D, why not L. But we will see L form of glycerol also present in different kind of species. But why it has not been actually determined, what is the basic reason? I think due to evolution that has been actually in the plasma membrane, this D form actually got an evolutionary advantage. That's why it has been incorporated. Okay, we will go into that. Why other species will have L form, why not D form? That answer has been actually answered. Okay, so plasmalogen, it is an exception in the so there are also enzymes, and very important is that why this kind of D form of glycerols are important, because important thing is that there are enzymes are present for D form. L form, there are no enzymes for L form present for during the metabolism. That's why D form of glycerol is present. Yeah. Now think very carefully. Come to the next section. That is about the heading though Archaebacteria. In case of Archaebacteria, in Archaebacteria they actually have L glycerol. Very important. They have L glycerol. They actually, this L glycerol bond with the fatty acid. With the fatty acid is actually the ether bond, and ether bond is like a very strong and stable bond. Now we actually know from our class 11 that stable bond in Archaebacteria, they actually live in hydrothermal vents like thermoacidophils. So there are huge amount of temperature. So they need actually very strong bond to maintain their plasma membrane. That's why the bond between the L-glycerol and the fatty acids, they are ether bond. That's why these ether bonds are not easy to hydrolyze. They are not easy to hydrolyze. So this is a very important portion. Questions are coming from in different exams. So L-glycerol is present in Archaebacteria and ether bond is present in the Archaebacteria. And which eukaryotic phospholipid has ether bond in the C1? Plasmalogen. We have also discussed that. Also certain important things I am going to tell you here, and other parts will be dealt in the microbiology section. So here they consist of, Archaebacteria consist of long chain fatty acids. Long chain fatty acids are actually present, about C20 to C30 carbon वाला fatty acids ये सब पाए जाते हैं. Long chain fatty acids are actually present, and most often the fatty acids are saturated fatty acids. We will be coming into the fatty acid parts in a while, don't worry about it. So most often the fatty acids are saturated fatty acids. Most often the fatty acids are what? Saturated fatty acids. And the double bond and the double bond present, if there is double bond. Normally there is no double bond if the fatty acids are saturated. If there is double bond, the double bond will be a trans double bond. What? Trans double bond. If there is double bond, it will be trans double bond, so that the structure remains very much stable. Okay, the structure remains very much stable. As simple as that. Okay. Now, so what we have discussed till, so we will just write a pictorial representation. So see this very carefully. Suppose this is the membrane. Sorry. Suppose this is the membrane. This is the membrane, and we are having different kinds of phospholipids. So suppose this is the membrane, and this is the outside, and this is the cytosolic phase. Outside core, we can also tell exoplasmic phase. Exoplasmic phase. Now, here this will be your okay. And another important thing is that you know that the plasma membrane is a double layered membranous structure, right? So now the thing is, these phospholipids which are actually present, okay, the phospholipids which I have mentioned, like phosphatidylethanolamine, phosphatidylcholine, these things, they are present in asymmetrical amount. So I am actually drawing this figure in this kind of thing. Okay, see this very carefully. And you just also draw with me. Just write it down with me like this. Now, what is actually this? See this very carefully. The phospholipid which is actually present on the cytosolic domain, this is, we can tell, PC, phosphatidylcholine, present on the exoplasmic, mainly present on the exoplasmic side. Phosphatidylserine is mainly present on the cytosolic phase, P, that's why it is called, we can also write it as PS. We also know that another phospholipid which is facing the cytosolic phase is PI, Another phospholipid which is also facing the cytosolic phase is called PE, phosphatidylethanolamine. Now, the kind of, these are all the phospholipids. Now, another kind of lipid, membrane lipid, which are present on the, mainly on the exoplasmic phase, it is a kind of phospholipid, but it is a sphingosine based phospholipid. We will be coming into the next portion. This is basically called the sphingomyelin. This is a sphingosine based phospholipid. Sphingosine based phospholipid. So you can see that there is a partial asymmetry. There is a partial, like 80% on the exoplasmic side, 20% on the cytosolic side, 20% on the exoplasmic side, 80% or 70% on the cytosolic side. Like this kind of arrangement. So we can tell that these phospholipids are having a partial asymmetry, in their distribution. another kind of, you can see this is another kind of membrane lipid, but you can only see it is facing the exoplasmic phase only. So there is no part of that membrane lipid facing the cytosolic phase. This is nothing but your glycolipids, mainly the sphingoglycolipids, sphingosine based glycolipids. So we can tell this thing is that the sphingosine based glycolipids, actually, as they are not facing any other phase other than the exoplasmic phase, so they are having total asymmetry. They are having what? Total asymmetry. And there is another kind of membrane lipid. It is actually not a lipid, but we actually learn in the form of lipid. This is also very important. So you can see that they are facing equally it is present in the exoplasmic phase, and equally it is present in the cytosolic phase, equally in the exoplasmic phase and equally in the cytosolic phase. So we can tell this is basically nothing but the cholesterol. And this cholesterol is very, very important. Cholesterol shows symmetrical distribution. It shows what? Symmetrical distribution. So which one shows total asymmetry? Glycolipids. Which one shows partial asymmetry? All the phospholipids. Which one shows only symmetrical distribution? That is the cholesterol. Is it clear, everybody? Is it clear? Very good. Next. Now give heading. Sphingosine based membrane lipids. So we have completed. Now, sorry. We have to note one thing. Very important. So we have actually completed what? Glycerol based phospholipids. Now there are also glycerol based glycolipids. So write it down. Chloroplast and thylakoid membrane. Chloroplast and thylakoid membranes consist of glycerol based glycolipids. These membranes only consist of glycerol based glycolipids, which are known as galactolipids, which are called galactolipids. That is the only information you have to know, not more than that. So which membrane consists of glycerol based glycolipids? That is the chloroplast and the thylakoid membranes. They consist of glycerol based glycolipids known as the galactolipids. So they are very less in amount, as they are restricted only to these chloroplast and thylakoid membranes. Now, next portion. Next portion is what? Is the sphingosine based membrane lipids. Sphingosine based membrane lipids. First, sphingosine based phospholipids. And only example you have to know is that sphingomyelin. It is an example of the sphingosine based phospholipid. Now what is the sphingosine and what was glycerol? These are nothing but the alcohol. So sphingosine is also an alcohol having N, having nitrogen, N group. So if I draw just a normal figure, I will not do the structure because that is restricted to biochemistry. I am not going to draw that. This is the structure of sphingosine, and there is N, and here basically what happens is that here one fatty acid is actually bind. This is the fatty acid, and this is what? This is the sphingosine. This is total, it is the sphingosine. Now what happens? Along with that there is a phosphocholine group. There is a phosphocholine group attached to this structure. So this is sphingosine, this is one fatty acid, and another is a phosphocholine group. What was present in phosphatidylcholine? There was a glycerol. There was a glycerol plus two fatty acids and having a phosphocholine group. Here what is present? Here is a sphingosine. Here is one fatty acid and a phosphocholine group. Phosphatidylcholine was present at the exoplasmic phase, mostly at the exoplasmic phase. And why? Because they are having a phosphocholine group. Sphingomyelin is also present at the exoplasmic phase mainly. So you can see that here also phosphatidylcholine is also present at the exoplasmic phase, having a phosphocholine group. Sphingomyelin, as it has a phosphocholine group, it is also facing the exoplasmic phase mainly. Okay. So here two fatty acids, here one fatty acid. Okay. Now important thing is that the bond between the fatty acid and the sphingosine, this bond is actually an amide bond. There was ester bond, here the bond is amide bond. Which kind of bond is present? Amide bond is present. Which kind of bond? Amide bond is present. Okay. This is also very important. This is also very important. Acha. If a question asks in this manner, like, see this very carefully, okay? Like this. What is this part? This is a phosphocholine part. This part means sphingosine plus this means sphingosine plus one fatty acid. It is called ceramide. This structural point we will go into in biochemistry. So actually sphingomyelin we can also write in this way, that sphingosine plus fatty acid plus phosphocholine group, and this we can also write as ceramide. So we can also write ceramide plus phosphocholine group. Very good. Is it clear, everybody? Is it clear? So please comment in the chat box. Comment in the chat box. Very good. Okay. Now up to this, tell me, are you liking the way I am teaching you? Are you liking the way I am taking the concepts in this manner? Thank you. Okay. Now let's go to the next part. That is actually the most important part. That is the sphingosine based glycolipids. Sphingosine based glycolipids. Very important part. Now, sphingosine based glycolipids, if you are going to deal with this, the thing is they are very abundant. I have also written in the first portion that is the most common glycolipids are always sphingosine based glycolipids. Okay, they show, I have also written here also, see this, I have also written here that the sphingosine based glycolipids show total asymmetry. Okay, they show total asymmetry. So I am just going to write it again so that you can revise it. So they show total asymmetry. They show what? Total asymmetry. They are present They are present on the exoplasmic. They are present on the exoplasmic membrane, exoplasmic phase of membrane, of the membrane. Now, see this very carefully. If a question comes like they are present on the lumen of Golgi body, is it correct or not? Is it correct or not? If a question comes that glycolipids are also present on the lumen of the Golgi body, tell me, is this statement correct? बोलो बोलो, क्या वो करेक्ट है? Just give me a random answer. Oh yes, it's correct. Yes, very good. So now the thing is, what is present? ये बहुत अच्छा concept mind में एकदम पूरा बैठा लो. That is also very important. I'm telling this. Suppose this is a cell, okay, and this is a particular, मान लो एक वेसिकल है, मान लो गोल्गी बॉडी का एक वेसिकल है. सो जो साइटोसॉल का जो एनवायरनमेंट है, सपोज दिस इज द एनवायरनमेंट ऑफ द साइटोसॉल, सपोज दिस इज द एनवायरनमेंट ऑफ द साइटोसॉल. थोड़ा सा ठीक कर लेते इसको, लाइक दिस. दिस इज द एनवायरनमेंट ऑफ द साइटोसॉल, राइट? Outside the lumen or outside the vesicle, it will have the same environment, and the environment which is present here on the exoplasmic phase, it will be present in the lumen of the vesicle, like this. Now you will say why? Because I will show you why. Because if you actually do like this, if there is a vesicle fusion, now do it like this. So you can see that this outer part, that is the exoplasmic part, that is actually present over here, which is going inside the lumen. So you can actually make a figure like this so that you can understand that what is present, what environment present within the lumen, that exact environment will be present on the exoplasmic phase, and what environment which is present on surrounding the lumen, that will be the environment present on the cytosolic phase. So that's why what environment present within the lumen, that same environment will be present on the exoplasmic phase. What environment present on the outside the lumen, that is the same environment in the cytosolic phase of the membrane. Yes, I am going to explain it again. See this very carefully. Suppose if I am having a particular vesicle, suppose this is a particular cell, suppose this is a particular membrane, and this is a particular cell. Sorry, this is a particular vesicle. This is a vesicle. Okay, this is a vesicle, and suppose this is a membrane. Okay, now double layered membrane, but I am just writing it for normally to make things easy. Suppose this is the cytosolic phase. Orange phase is the cytosolic phase. So you tell me one thing, the surrounding of the vesicle should have the same environment like that of the cytosol, because it is actually facing the cytosol, this outside. Is it not? It will be. Now what happens if I actually draw the exoplasmic phase environment like this? It is said that the inner portion of the vesicle, that is the lumen of the vesicle, will have the same environment which is the environment of the exoplasmic phase. Now you will tell why, sir? Sorry, why Shwedidda? Because it is happening like this. Suppose if you want to fuse the vesicle with the plasma membrane, so there will be a vesicular fusion, and when there will be a vesicular fusion, so you see that it will become like this. So it will fuse with the vesicle, and as it fuses with the vesicle, so you can see that what is present outside the lumen will have the same environment like that of the outside the lumen, like this, and what environment which is present inside the lumen is present at the exoplasmic phase. Now is it clear? I think it's clear now, right? Very good. Okay. Now write one thing. Is that note. Make a note. These sphingoglycolipids have a broader head and shorter tail. What? Broader head and shorter tail. These sphingoglycolipids have what? Broader head. Because why they are having broader head? I am telling you. Broader head and shorter tail. Why so? Why? Because they are having glycolipids. They are glycolipids. These are the bloody fatty acid chains. That is fine, not a problem. These are the polar heads. Now along with that there are multiple sugar associated with this. Multiple sugars are present. So this is, suppose this is a particular lipid, and if you are having multiple sugar associated with this, multiple sugar. So from outside you can say that they are having a broader head and a shorter tail. They are having what? Broader head and shorter tail. Why broader head? Because they are having multiple sugars associated with them, as they are a glycolipids. So multiple sugars are associated. Now what happens? See this very carefully. So as a result of that, what the thing is going to happen, as they are having broader head and shorter tails, they are not able to form a bilayer structure. Means sphingoglycolipids alone are not able to form lipid bilayer, as they are having, please write it down, as they are having large head and small tails. So if you create a scenario that only sphingoglycolipids are present, they are not able to form a bilayer structure. No, it's not possible. So what is very important is that suppose if you place simple phospholipids, and if you actually pour them in the water in a particular concentration, they will form a bilayer structure like this. But if you actually pour only sphingoglycolipids, so they can't actually form a bilayer structure. So now what happens? So they have a particular affinity for another kind of a lipid which has the exactly opposite form. And what are they? Now comes the concept of cholesterol. Cholesterol have small head and larger tails, and these sphingoglycolipids have a huge affinity for cholesterol. Why? Because they are having the opposite thing. Huge affinity for cholesterol. Like they are having larger heads and smaller tails. And they need somebody with smaller heads and larger tails. So that they can actually associate in a proper manner to form a bilayer. So in order to form a bilayer, so sphingoglycolipids, yes. Cholesterol, in the structure of cholesterol has smaller head and longer tails. Sphingoglycolipids have smaller tails and larger heads. So they are actually complementing each other for a particular binding. That's why if you place cholesterol along with the sphingoglycolipids, then actually you can form a biliary structure only with those two things. And as a result, we can see that cholesterol and sphingoglycolipids, they have a huge affinity for each other because they actually in structurally they complement each other, so that the total binding efficiency in a biliary form is actually perfect. That is the main concept. So sphingoglycolipids have higher affinity for cholesterol in order to, and if these two are present, then actually they can form a bilayer. Okay, so please take a 1 minute break. I'll be joining you in 1 minute. Okay, in 1 minute, just take a 1 minute break. Take 1 minute break. Just 1 minute. In 1 minute I am coming. Now, see these things. So we have completed what? We have talked about these things, these glycolipids, sphingoglycolipids as well as the cholesterol. Now, the thing is, which is actually, I'm just actually revising it very clearly. So we have talked that the plasma membrane which is present, I have drawn the plasma membrane like how I have drawn this like this. So how it is present, see this very carefully. We have a lipid layer mainly, and how it is present, see this very carefully. So plasma membrane is, we know that it is a bilayer structure. Okay, it is a bilayer structure. I have explained you individual components. Now I am actually summing up all these things. Okay, now what is present, see this very carefully. These are the lipid layers. This is the lipid layers, which are mainly the phospholipids, which I have mentioned, all these kind of things. Now very important, these are normally the phospholipids present, and this is how the bilayer structure is present. Now the thing which I have also mentioned over here, that this phosphatidyl choline, phosphatidyl serine, phosphatidyl inositol, phosphatidyl ethanolamine. Now how it is basically present in the exoplasmic phases or in the cytosolic phases? First of all, think in this manner, like that the number of phosphatidyl choline, it's the concept lies here. There are many interview questions are Shuridhar, we say that the membrane with the sphingosine glycolipid will have more rigidity as cholesterol is present. Yes, I will come to that. I will come to cholesterol. Yes, no, it will not make the membrane more rigid. Actually, what happens is that it will maintain the rigidity. It will not lose its rigidity. It does not actually, sometimes it creates a rigidity, but the main function of cholesterol, I will be coming into that in a few minutes. Just explain me this topic. So these are actually the main, what is actually thinking. This is the double membrane, this is the lipid bilayer, and sometimes you can see that these phospholipids are actually the number of phosphatidyl cholines or the number of sphingomyelins. These lipids are actually in number facing the exoplasmic phases much more than the other phospholipids. On the other hand, phosphatidyl serine, phosphatidyl inositol, phosphatidyl ethanolamine, these phospholipids are mainly facing the cytosolic phases in more number. Okay, so the thing is, you have to know that the membrane, this plasma membrane, is a bilayer structure of the lipids, and these membrane lipids in facing the exoplasmic phases is more number. Now I'll make you see some videos, so please be with me and tell me that you can see the video or not. Tell me that you can see the video or not. This is a 3D view of the membrane. Just see that. Okay. So basically you can see that this is the structure. These are the basically these are mainly these are the phospholipid structures. These are the phospholipids. These are the lipid layer which are present. So you can see that these membrane is present in a bilayer format. These are present in a this is one layer, this is one layer, and this is another layer. So these, you can see that these are the bilayers of the lipids which are present. Okay, now these are the lipid bilayers, and this, it is the overall upper view of the membrane, and you can see that there are multiple proteins are are present inside that of the membrane, which are coming out, which are having these things which are actually like this. These are actually the sugar coating. These are the glycoproteins. There are certain glycolipids are also present. Okay, these are the proteins, these are the proteins, these are the proteins, and all these things which you are seeing, these are the polar heads. These are the polar heads of the lipids. These all are the polar heads of the lipids. This is a 3D view. This is a 3D view of the total membrane having a bilayer format, and which I have told that these phosphatidylcholine, phosphatidylinositol, all these kind of things, these are actually what? Different kinds of membrane lipids. Okay, so the number of phosphatidylcholine on the exoplasmic phase are much higher than that of the number of other phospholipids. That's why we are representing in that format that number is high on the exoplasmic phase. That is the main concept, and these are actually the different kinds of phospholipids, and these are the membranous proteins. These are the proteins. Okay, these are different kinds of membranous proteins. These are also different proteins present in the membrane. So these are actually a 3D representation of the membrane. So these are the proteins which are being glycosylated. Okay, these chains are actually, these are the sugar residues. These are the sugar residues. Okay, these are actually the sugar residues. So you can see that this is the total overall structure of the membrane. So this is one short view of the membrane. So if we actually come to another figure, so we can actually see that this is the overall structure of the membrane, and you can see these are actually the different bilayers. So why I am telling you this, because I have explained to you all these kind of lipid portions, and I am just making you see what is the actual structure of the membrane. So these are actually the phospholipids layer, and these are actually, you can see that green portions, green layers. These green are the glycolipids present over here. There are certain kinds of protein which are present. We'll be dealing with the protein in the later classes, not today. Okay, certain amount of cholesterol. See that there are certain amount of cholesterols are also present, and you can see also that the cholesterol are actually symmetrically present. Okay, and all those phospholipids which I have mentioned, there are partial asymmetry in those lipids. So this is the overall structure of the plasma membrane. So this is also, you can see that this is a bilayer structure. If we divide, these are the two layers, like outer phase and the inner phase, and these are present like this manner. So this is the overall structure of the plasma membrane. Now let's come to another important concept, that is the cholesterol. So let's come to another important concept we have done so far. So now we will come into another important concept called the, before going into that, we have also one important thing. See this very carefully. In glycolipids, heading go, sphingoglycolipids. In sphingoglycolipids, what is actually important is that there are of two types. Sphingoglycolipids are actually of two types. Now I am going to into a deeper level, and I am going to actually cover many concepts regarding a little bit concept of viral entry and viral exit also. So be with me very cautiously. This is very important portion. First, sphingoglycolipids can be one of cerebrosides. When it is called cerebrosides, if single sugar is attached, whereas in another kind of sphingoglycolipids is called the globo, as well as there are other kind of called globocytes and gangliosides. So globocytes and gangliosides. But out of that, and why they are called globocytes and gangliosides? Because there are multiple sugars are attached. When multiple sugars are attached, when oligosaccharides are attached, then it is called globocytes. When polysaccharides are mainly attached, they are called basically the gangliosides. So multiple sugars are attached. Multiple sugars attached. Now see this very carefully. Very carefully. Now example of a cerebrocyte is glucocerebrocyte. If glucose is attached with the sphingolipid, okay. Glucocerebrocyte is an example. Glucocerebrocyte. Okay. So like this is the sphingosine. This is the sphingosine. This is a fatty acid, and in the sphingomyelin there was a phosphocholine group. Now here there is nothing but a glucose. This is the glucose, and the bond between the glucose and the sphingosine is basically a glycosidic linkage. This is the glycosidic bond, and this bond is actually, we have told about this is an amide bond. Okay. Now note one thing. Suppose if in the lysosome if glucocerebrosidase enzyme is absent, if in the lysosome glucocerebrosidase enzyme is absent, then in the lysosome glucocerebroside will not be degraded. Glucocerebrosides will not be degraded, so there will be accumulation of the glucocerebrosides in the lysosome. There will be what? Accumulation of glucocerebroside. There will be accumulation of glucocerebrosides in the lysosome, and this leads to a particular lysosomal storage disease. It leads to a particular lysosomal storage disease, and the name of the disease is also very important. Is called the Gaucher disease. Many questions come from this. Gaucher disease. Gaucher disease is a kind of lysosomal storage disease when in the lysosome glucocerebrosidase enzyme is not present. As a result of that, glucocerebrosidase accumulation takes place in the lysosome. That's why it is called the lysosomal storage disease. Okay, it's an autosomal recessive disease. You will be dealing with that in the genetics lecture. That is also very important. Out of those globocytes and gangliosides, most common are the gangliosides, and these things, gangliosides, are very, very important. Yes, Shuman, I will repeat. I have just told you that one of the disease related to this glucocerebroside is that sometimes in the lysosome consists of different hydrolytic enzymes, right? So if the glucocerebrosidase enzyme, which actually degrades the glucocerebrosides, this enzyme is absent, then what happens? There will be no enzyme to degrade these glucocerebrosides, so there will be more and more accumulation of the glucocerebrosides in the lysosomes, and as a result of that, a lysosomal storage disease will occur called the Gaucher disease. This glycosomal storage disease is an example of an autosomal recessive disease. It's an autosomal recessive disease that will be dealt in the genetics lecture. Now let's come to ganglioside. So did you understand Shomana? Okay. So now gangliosides, they are also very important because ganglioside consists of one or more sialic acids. Sialic acid is a kind of sugar. It is also known as sialic acid is the kind of sugar called N-acetyl neuraminic acid. It is called NANA. in this, from this section, huge amount of questions are coming in interviews as well as in different, and as there is a COVID-19 pandemic, maximum questions can come from this section. Why? I'm going to tell you that. Okay, so these sugars are present, N-acetylnuraminic acid or the sialic acids are present in the gangliosides. Okay, suppose if one sialic acid is present, such kind of gangliosides are called as GM gangliosides. Such kind of gangliosides are called GM gangliosides. Mean gangliosides having monosialate condition. Okay, and there are different varieties of this ganglioside monosialate condition like GM1, GM2, GM3. All these kind of different types of GM gangliosides can be present. GM1 gangliosides acts as the receptor for cholera toxin, E. coli endotoxin, and influenza virus, and there your comparative level questions can arise. See this very carefully. Now what happens? See this very carefully. What happens? In several viruses, there are two kinds of marker proteins. Suppose in influenza virus, this is very important because influenza virus has the same topological domain that of the coronavirus. Coronavirus though are huge, the structure is much higher, much larger, but both of them are positive sense RNA virus, and this thing is also mainly common. What happens? Influenza virus has a marker protein. Called hemagglutinin. Please check the spelling ekbar. And neuraminidase. See this very carefully. What happens? You know that in our cell there are several sphingoglycolipids and there are certain kinds of gangliosides. Hemagglutinin actually recognize actually recognize the sialic acids, of the gangliosides present in our membrane, and different cells have different kinds of gangliosides. Not all sialic acids will be actually recognized by the hemagglutinin. Specific sialic acids will be recognized which are present in separate cells or specific cells. So suppose this is a virus, and in the virus there is a particular marker protein called the hemagglutinin. They will actually recognize the sialic acid residues in the gangliosides, and as a result of that it will bind with the ganglioside sialic acid, and as a result of that there will be a vesicle fusion and the virus will enter. So this will lead to the viral entry. It will lead to what? Viral entry. Now suppose that a virus has entered into the cell. Now what is happening? Is that it needs to go out of the cell, and for that it needs to cleave these sialic acids. And I have told you this sialic acid is nothing but the N-acetylnuramic acid. For that, neuraminidase will play an action. They will cleave the sialic acid. What they will do? They will cleave the sialic acid, cleave nana or sialic acid, and that is actually important for your viral exit. That is important for your what? Viral exit. Important for what? Viral exit. So hemagglutinin recognizes the sialic acid for viral entry. Neuraminidase cleaves the sialic acid for viral exit. So that's why what is happening is that in influenza virus has specific kinds of H, hemagglutinin and neuraminidase. Influenza virus which has the H1N1 strain. Okay, they are actually pathogenic for the humans. So H1 and N1 actually recognizes such kind of sialic acid which are present in our cell membranes, gangliocytes. So they are pathogenic for human. And H5N1, they are pathogenic for birds. And that's why these gangliocytes are very much important for different markers in the cell. So these glycosilipids acts as a markers in the cell, as well as these phospholipids does not act as a marker in the cells. Am I making my point? Everybody clear now? It was a very good concept. Clear? Tell me everybody, is it clear or not? Tell me everybody, is it clear or not? Okay. Now, another concept. Now, let's come to another concept, that is the fluidity of the membrane. Given heading called the fluidity of the membrane. It's a very easy topic, you can actually easily understand all these kind of things. It is an easy topic. See this. Fluidity of the membrane is mainly due to the membrane lipids. Is mainly due to what? The membrane lipids. And these actually lipids and this fluidity is actually depends upon the type of fatty acids. They actually are dependent upon the type of fatty acids. Now, can you tell me what is TM? What do you mean by TM? Please comment in the chat box. What do you mean by TM? What do you mean by TM? Please comment in the chat box. What is TM? Okay, so Preeti, tell me where there is DNA. So I am not talking about DNA. So in this scenario, what will be the TM? It will be a thermal melting point. Yes, it will be thermal melting point, but TM is not always related to DNA. Okay, it's related to everything. Thermal melting point. Now, so it is the temperature at which the membrane lipids are at 50% in gel form and 50% in sol form. Sol means totally in liquid phase, gel means in totally a gel like phase. So gel and sol. So the temperature at which 50% of the membrane lipids is actually gel form and 50% of the membrane lipid is actually in the sol form. And the instrument used to check the thermal melting point is also very important. Questions are coming in JNU. Instrument used to check TM, that is differential, very important, scanning calorimeter. DSC. This is the instrument which are used in order to calculate the TM, right? So like suppose if I draw a graph like this, suppose if I want to sol state, this is the solution state and this is the temperature. So I am increasing the temperature. At first it was present in a gel form. So it was present in a gel form, and after that it has formed in a solution form. So it was in the gel phase, and now it is in the sol phase. So now this kind of graph is called a sigmoid graph. We will be dealing in all these kind of things in biochemistry. Sigmoid graph. Why? Because it is actually occurring in a cooperative manner. Means, suppose you are giving a temperature and all the lipids are being melted, and they are actually happening in a cooperative manner. Individual lipid melting, that is not happening. Actually the lipid is melting in a cooperative manner, and that's why the gel state is actually converting it into the sol state. So at the temperature where there is 50%, the temperature at whether there is 50% gel state and 50% sol state, that will be your thermal melting point, or the TM. So that is an important concept you have to know. So now this TM, this thermal melting point actually depends upon the nature of fatty acids. What? This thermal melting point depends upon the nature of fatty acids. Depends upon what? The nature of fatty acids. Point number 1, we have to know that this thermal melting point is directly proportional to the fatty acid chain length. It is directly proportional to the chain length of the fatty acids. It means that if we have a fatty acid with higher chain length, the thermal melting point will be more. If we have a fatty acid with lesser chain length, it will have a lower thermal melting point. Okay. And second most important point is that the thermal melting point is inversely proportional to the degree of unsaturation. Degree of what? Degree of unsaturation. Mainly it is a cis bond. Mainly bond which are occurring, that is the cis bond, because trans bonds are very much less in the natural system. Okay. So suppose if we have a fatty acid, I will discuss detailedly in the biochemistry section. Suppose if we have a fatty acid like stearic acid, I am giving you all the details, you just tell me. Stearic acid is 18 carbon, and 0 unsaturation. And we are having oleic acid, that is also 18 carbon with 1 unsaturation. So this is A and this is B. Now tell me in the comment box which fatty acid will have higher melting point, A or B? So I got one answer. Tell me everybody. I got 4 answers. Tell me everybody. Right. Why? Because stearic acid doesn't have any unsaturation. That's why it will have a higher melting point. Now, suppose if I go it like this, suppose if I give you a question like this, we are having DOPC, okay, diollyl phosphatidylcholine. Diollyl phosphatidylcholine, 1, it is a kind of phosphatidylcholine. Second, we are having stearyl ollyl phosphatidylcholine, and C, disteryl phosphatidylcholine. So out of these three, give the order of TM. Give the order of TM. So is it 1, it is 2, it is 3. What will be the order of TM? Diollyl, stearyl ollyl, and disteryl, because PC is common. There is nothing to do with the PC. So okay, so there are 2 kinds of answers. There is 1 3 1 2 and there is 3 2 1. So let me clear that out. See this very carefully. First of all, that in case of disteryl we are having 18:0, 18:0, right? Here we are having 18:0, 18:1, and here we are having 18:0 and 18:1 is 18:1, 2. So it is evident now. It's evident there is 0 unsaturation. So the thing is, where there is least unsaturation, there will be more Tm. So 3 will be more greater, and then there is 1 unsaturation, okay, and 0. So as only 1 is unsaturated and other is not unsaturated, that's why 2 will be, and then it will be 1. So order will be 3 to 1, not 3 1 2. Okay, that is the case, right? Now another important point, you have to know that if, now also write a note, adaptations to, suppose a particular organism is actually adapted to a low temperature. Now what will be the profile of the fatty acids in the plasma membrane of the cells? So adaptations to a low temperature, if there is, so what are the adaptation to low temperature? For low temperature you need short chain fatty acids, and unsaturated fatty acids. Now there is a very important concept and marvelous concept in this, but I am not going to explain this because it is a totally concept of the lipid portion of the biochemistry. I am just mentioning the important facts and I am going out of the membrane lipids, okay? And what are the important adaptations? Actually there is a concept regarding melting point, freezing points, awesome concept, but I will be telling you in the biochemistry section, so no need to worry about it. And what are the adaptations to higher temperature? Just opposite, long chain fatty acids and saturated fatty acids. This is also very important, and that's why I have told you in archaebacteria you are having long chain fatty acids and saturated fatty acids. And what kind of bond? Tell me in the comment box, what kind of bond is present in archaebacterial membrane? In the membrane lipids of the archaebacteria, what type of bond is present which is actually not easy to hydrolyze? Yes, ether bond. It's very important. It's the ether bond. Now let's come to sterol portion. Let's come to the sterol portion. Sterols. So there are many kinds of sterols. We'll not go into the detailed structure, that is not essential, but I'll mention few names which are actually important. A. Cholesterol. Cholesterol. This is unique to animals. Cholesterol is unique to animals, but there are other kinds of sterols also, like brassicossterols, like cetosterols, like stigmasterols. Why these are important? These are important because these are the sterols present in the plant cell membrane. These are called the phytosterols. Phytosterols are those, these are those sterols which are present in plant cell membrane. Which are present in what? Plant cell membranes. Another important sterol is also present. This question has come in BHU PET, ergosterol. Ergosterol is present in yeast. Ergosterol is present in yeast. Important point. Note. What is an important point? In prokaryotes, sterols are actually absent. In prokaryotes, sterols are actually absent. But sterol-like molecules can be present. And they are called the hopanoids. They are called what? Hopanoids. Hopanoids are sterol-like molecules present in the prokaryotes. These are also very important. Now let's go to the functions of the sterols. function of sterols. We will be mainly dealing with the cholesterols, function of sterols. So regarding the membrane portion, not the physiology portion. Physiology portion will be dealt in the biochemistry section, but we will be only dealing with the membrane or the cell biology portion. First point, as Bivas told me, that it maintains membrane rigidity. Maintains membrane rigidity. I will give you a good concept regarding this. Just stick to it. Point number 1. Sterols act as a, it's a very important statement, they act as a temperature buffer. They act as a temperature buffer. Means if you actually change the temperature higher or low, membrane configuration will remain constant. So it is acting as a temperature buffer. We have pH buffers. If you change the pH, the buffer will maintain the pH. They also change the even if you change the temperature, they will maintain the condition of the membrane. Whether you change the temperature, they can't change, they can maintain the temperature. First of all, sterols will not maintain the temperature, but if you change the temperature, they will actually maintain the fluidity of the membrane. That is an important concept. That is an important concept. Now what is important again? See this. Third point, they also do not allow the lipids to aggregate at low temperature. If you create a low temperature, if you give a low temperature, it will act as a temperature buffer and it will prevent to lipids to aggregate, so that the fluidity remains constant. So we can tell that it maintains the fluidity of the membrane. It also maintains the rigidity and also it maintains the fluidity of the membrane. It also maintains the fluidity of the membrane. That is also very important concept. So basically what is actually it's doing? If you give temperature, there will be transition from gel to sol form or sol to or if you take out the temperature, or if the temperature is lowered, they will be converted from sol form to gel form. But if sterol is present, if cholesterol is present, what will happen? They will try to abolish the transition. They will try to abolish the transition. That is also very, very important point. They will try to abolish the transition. And we know that cholesterols, we have also studied, they are symmetrically distributed. They are symmetrically distributed over the plasma membrane. Symmetrically distributed. The numbers are symmetrically distributed in exoplasmic as well as the cytosolic phases. That is also very important. That is also very important. So now it is a question of the interview. One interview. I'll give you a question and you just tell me what will be the answer. Suppose if we are having a graph, it's a fluidity versus temperature graph. Okay, so now it's 12, it's 2, 22, okay, like 32, 42, 52, 62, like this Celsius. Now when there is no cholesterol, if you increase the temperature, the fluidity will go from gel form. This is the gel form. It is the gel form to the sol form, right? So you can see that at 22 degrees Celsius it is gel form, and at about 52 degrees Celsius it is the sol form. It is what? The sol form. Now if you add, so this is the condition when there is no cholesterol. So what will be the graph what will be the fluidity versus temperature graph if there is cholesterol? then what will happen? See this very carefully. They will actually abolish the transition and they will try to maintain it in a similar phase, though there is a change in the temperature. But after a lot of change, they can't do anything. So what happens? They will try to maintain it in a similar condition. But after that, suppose from 12 to 62, they will try to maintain in a similar condition. After that, they can't. Then it will be converted to sol form. And again, if you decrease the temperature up to 12 degree or 2 degree, or this is 5 degree, it will try to maintain in a same condition. But after that, if you lower the temperature so high, it will come down like this. So a better example will be like this. It will be like this. Okay, after that it will come down like this. So it is the condition when if cholesterol is there. Why? Because you can see that if cholesterol is there, you can see that here in 22 you are having gel form, in 52 you are having sol form, but here even in 12 you are not having a complete gel form, even in 10 you are not having a complete gel form, in 2 you are having gel form. Similarly, you are not having a complete sol form in 52, even 62. After that, above 62, you are getting a sol form. Yes, I am repeating once again. See this. What happens is that in this graph, it has been explained is that they will try to maintain what was the actual maintain the fluidity of the membrane. You increase the temperature, normal condition. If there was no cholesterol in 52, you are getting the sol form. All the lipids has been melted. And at 22 degrees Celsius, all the lipids has been freezed. There is gel form. But if you add cholesterol, they will try to maintain the lipid fluidity which was present normally in the optimum condition, that was the 42. It will try to maintain in this phase. But if you add more temperature, it will again try to maintain in 52, again try to maintain in 62. But after a huge amount of change in temperature, it can't. Then it will go into sol phase. If a huge amount of low temperature if it's there, then it will go into gel phase. But for a large period of time, so can we describe an increase in the relation to the kinetic energy of the particle after a particular temperature? Yes, obviously, because there is a particular kinetic energy increase if you increase the temperature. But those cholesterol try to prevent such the molecular Brownian motion. But after a huge amount of change, the kinetic energy will actually surpass the this cholesterol action, and there will be phase change. Yes, you are absolutely correct. So everybody clear to this point? This is a very important concept. Everybody clear? Okay, so today this was mainly all about, and I will just add the last section is that last portion is that another kind of lysosomal storage disease you have to know, that is called the if in lysosome sphingomyelinase enzyme is absent, then what will happen in the lysosome? There will be accumulation of sphingomyelin, and it will lead to a lysosomal storage disease called Niemann-Pick disease. This is also a lysosomal storage disease. That is also very, very important. Lysosomal storage disease. Okay. So this was all about today's class. We have actually dealt with all the membrane lipids portion. Next class we will go, those who will join our lectures, my class will be on the next week, and the second demo class, I'll tell you the second demo class will be on 29th July. 29th of July, that will be our second demo class on biophysical methods from, are you actually available this time? Is good, 3 to 5:30? Is it good? Tell me in the comment box. Yes. So the second demo class will be on 29th of July and the third demo class will be on 31st of July. So we will know the date, we will give you the date or the time on the 29th, and I will send you all the important links regarding this second demo class. We will send you from our sales number. Okay. Yes. So tell me one thing. Did you like today's class? Did you have any doubts regarding today's class? Okay, from August we will change the schedule, don't worry about it. This is just a demo class. These are the all demo class. My class will start from the next first week of August and it will start from this portion. Membrane lipids is done and we will start with lipid grafts and membrane proteins. So tell me, everybody clear? Today's class was good or bad? Tell me, please feel free to give your opinions. It means a lot for us. Thank you, Priya. Everybody please give their opinions. Okay. Yeah, actually it needs to be informative because you need to know all these kind of things. It's very important. That's why we are actually focusing on important things, that when we are actually teaching the class, please make your notes. If you only see the class, that is not going to, yes, you have to make your notes. Thank you students, thank you very much. Thank you juniors actually. Yeah, thank you. It was wonderful to take you the class. Meet you on 29th from 3 p.m. and 2:45 will be the joining time. Okay, chalo, bye bye.